All right, today we're going to be talking about chemical reactions and enzymes. So, what exactly is an enzyme? Well, an enzyme helps in a process that changes one or more substances into another. Okay, and that process is called a chemical reaction. You may have heard of that from your middle school, chemistry science that you went over. So some of these reactions can occur really, really slowly, such as rust on a bicycle. It takes a really long time for that rust to form. Sometimes it's really fast. So like an atomic bomb is a chemical reaction, and once it starts, you cannot stop it. It occurs very, very quickly. So what is a chemical reaction? So chemical reaction has two different parts to it. It has reactants and it has products. So reactants are the things that cause that chemical reaction. Your products are made because of that process of the chemical reaction. And that happens all the time in our bodies. If you just ate breakfast, because it is morning time as I'm recording this, your body is going through chemical reactions to break down the foods that you're eating. So that way, they can build products off of that food, especially if it's something nutritious. So some reactions involve breaking bonds in the reactants and then forming new bonds in the products. So a very good example, again, if we think about what we eat, we're not made of, bread. It's not like your arm is a loaf of bread. It's not like your stomach is a steak. Essentially what ends up happening is whenever you eat food, all the bonds that are in that food and those biomolecules are broken down and they're used to form new things, such as whenever you have a cut, you have to repair the skin. If you have a bruise, your blood cells have to break down, the coagulated blood that's part of that bruise. And so you're always forming new cells. And so we're going to be using the foods that we eat in order to break those bonds and recreate them into whatever it is that our body needs. So they also involve changes in energy. It takes energy to digest our food, and then we also need to store up energy just for everyday processes. So chemical reactions that release energy occur spontaneously. Okay, so it just happens. And so, there's no rhyme or reason. It's just going to happen. We consider that spontaneous. But chemical reactions that absorb energy need some sort of energy source. You need to supply energy for that reaction to occur. So, for example, okay, so cellulose in your paper, it's going to release energy. Okay, so when we're talking about releasing energy, we're talking about burning something in the presence of oxygen. We're also talking about breaking bonds. So, will it light on fire now? Like if your paper is just sitting on the desk, is it going to light on fire? No, we need to give it energy. We need to get it energy in order to start up, and that's called activation energy. So activation energy is the energy needed for a reaction to begin. Okay, so it's energy needed to activate a reaction. That's why we call it activation energy. So where do enzymes come into play with this? Okay, so enzymes are types of protein. Okay, so we talked about proteins causing chemical reactions. And so when we're talking about that, we're referring to enzymes. So enzymes are proteins that are also known as a catalyst. Okay, so a catalyst is something that speeds up a chemical reaction or causes a reaction to occur. And so enzymes' roles are to speed up chemical reactions that take place in cells. And so how that happens is it lowers the activation energy. So if I go back to this chart right here, so I can see that I have activation energy right here, okay? So that EA is my activation energy. So that green line is without an enzyme. So if I had that piece of paper sitting there, maybe eventually it'll catch on fire. I don't know, probably not. So if I added an enzyme, okay, added something that lowers that activation energy, I don't need as much energy for that reaction to occur. And I'm still going to get the same product. So, okay, you have your initial state, which is where your products are at. Your final state, okay, is where your initial state is where your reactants are at. Your final state is where your products are at. So, what do you understand? An enzyme lowers activation energy, which speeds up the rate of reaction. And what type of biomolecule is an enzyme? Very good. It's a protein. Excellent. So one of the things we need to know about enzymes is that they are reusable. It'd be terrible if our body used up an enzyme once and then we'd have to make new ones. We'd be forever burning energy just trying to make these new molecules. So enzymes are reusable. So for example, okay, one catalase is a type of enzyme, one catalase molecule can convert 40 million molecules of hydrogen peroxide to water and oxygen each second. Okay, so catalase is an enzyme that's particular to hydrogen peroxide. It will break down hydrogen peroxide. So our body actually naturally produces a little bit of hydrogen peroxide through our metabolism, and hydrogen peroxide is actually toxic to us. But if you look at the chemical formula of hydrogen peroxide, it's H2O2. It sounds a lot like water, right? And so that's what catalase does. It breaks it off into water and oxygen. So some characteristics of enzymes, like what does it even look like? Okay, they're very specific. Catalase only works on hydrogen peroxide. There are other types of enzymes. Amylase, we have that in our spit. It works on starch. That's why if you put a potato chip in your mouth, it's going to start to dissolve if you let it sit there. Lipase works on lipids. We find that in our stomach. So our stomach's going to be breaking down lipids. Same thing for protease. Protease breaks down proteins. That's also found in our stomach and our small intestines. So each enzyme binds to a specific substrate. Okay, so and that location is called the active site. Okay, so enzymes are usually named for whatever their substrate is, and it ends in that suffix ase. Okay, so ase is added to the end of that word. Cellulase breaks down cellulose. Lactase breaks down lactose, which is a type of sugar. Protease breaks down proteins. Catalase breaks down hydrogen peroxide. So how in the world do we make this product? Okay, so we have our enzyme, and then we're going to start with the substrate. Okay, so I have my little example right here. Okay, here's my enzyme. Kind of looks like Pac-Man. Okay, the inside right here, this is my active site. I'm going to have a substrate that fits perfectly into that active site. Okay, so you can see, okay, my substrate, okay, it fits right in, okay, perfect fit. So my enzyme is going to work on this substrate, and when it's working just like this is called an enzyme substrate complex, okay, it's together, enzyme substrate complex. So after my enzyme does its work, it's going to be broken down, okay. So now, okay, it's worked, it's done its thing, so now I have a product, okay. So in this case, my substrate, okay, which started out whole, I know they're two different colors, is now a product, okay. So in this case, my enzyme broke down my substrate, and I can see my enzyme is unaffected, it's ready to start again, okay. I can have a new substrate come in, it's going to do the exact same thing, okay. I could also have the opposite, where maybe I have two substrates fit in, okay, and now all of a sudden, okay, it's put them together. All right, so I can have it where it breaks down or where it builds up. So decomposition, okay, that's the fancy word for breaking something down. Okay, that's where I took my substrate, okay, put it in my enzyme, made my enzyme substrate complex, and I got my two products. So there's a couple things that can impact whether or not an enzyme is going to work. Remember, an enzyme is a protein. Okay, so proteins are made of our amino acids. And so there's several different things that, you know, can affect your protein, and we call that denaturing. Denaturing is where you permanently change your protein shape so it's not going to work. So, you know, the reason why denaturing is important is because it can happen to us. We're primarily made out of proteins. So anything that's going to affect the normal chemical reaction, you know, is going to affect whether or not this enzyme is going to function. Okay, so some examples, pH. Okay, if you have something that's too acidic or too basic, it's going to impact this. So if you remember, when we talked about amino acids are made of carbon, hydrogen, oxygen, and nitrogen. pH means percent hydrogen. So if you're changing the percent of hydrogen, then that means that you're going to have more or less hydrogens on this enzyme, and so it's going to change its shape. So also the amount of enzyme or substrate. Okay, so usually if you have a substrate in the solution and you increase the number of enzymes, the reaction could happen faster. Eventually, if you have a set number of enzymes, it doesn't matter how much substrate you add because all the enzymes are working. It's not like an enzyme can work on two substrate at once. Temperature. Temperature is another one. Okay, so if you think about an egg is a type of protein. So whenever you cook an egg, it becomes solid, becomes a solid mass. Same thing could happen to your protein. If you cook it, it's going to change its shape, okay, and you may not be able to rechange it back. Same thing if it gets too cold. So most of our enzymes work at 98 degrees Fahrenheit, and that's because that's our body temperature. So if we get a fever, our enzymes are not going to work very well. If our temperature goes too low, same thing. So if there's an extreme change in temperature, the enzyme is going to be denatured, okay, and that's another word for destroyed. We can also have inhibitors. Inhibitors compete with the substrate, okay, so I know normally my substrate comes in here, okay, and it's going to fit right in. I can have an inhibitor, okay, so something, it doesn't fit properly, okay, but it fits well enough that it's going to go into my active site. And so I can see because my active site doesn't perfectly bind, it's not going to react to this, but this is blocking the substrate from going in. That's an inhibitor. A lot of medicines work like this. They inhibit the enzyme from working. So I could also have an external inhibitor. Okay, so I know my enzyme normally looks like this, looks like a letter C. I could have an inhibitor that binds on the outside, okay, just like that. Okay, and it's going to change the enzyme shape. So it's going to close my active site, so now my substrate can't go in. So there's a couple different types of inhibitors. Enzymes also can work together, and this is what happens in our body. You have multiple enzymes that work in an assembly line, and it's going to speed up chemical reactions in your body. So if one of those enzymes gets denatured, it's going to stop the entire process.